Hi everyone, and thank you for being here. I'm Karine Gemeni, a research fellow from the University of Würzburg, and I'm very happy to be here again to moderate this SONA webinar. And today we have a special guest. This is Dr. Eric Perkins, who is the Director of Product Management at Addgene. I hope you guys remember we had this summer Dr. Joanne Karens who talked a little bit about Addgene and her own resume, how she got into this non-profit organization. And I am sure today Dr. Perkins is going to give us more depth or in-depth into what Addgene actually does. So this webinar series is a joint initiative between the Society of Neuroscientists of Africa, Trend in Africa, the African Society of Neurologists, and the International Parkinson's Disease Genomics Consortium, Africa. With this initiative, we aim to provide a great learning and exchange platform, mainly to neuroscientists and to everyone interested into neuroscience. And at the same time, this is a great opportunity for younger fellows to get mentorship from experts. So I really want to say a little bit about Dr. Perkins. He received his PhD in genomics and molecular biology from UNC-Chapel Hill in the USA, where he studied DNA repair and VDG recombination. And after a postdoc that he spent studying DNA replication in Harvard's Department of Molecular and Cellular Biology, he started his career at Addgene in 2008. So this is already like 12 years now. He started as a technical customer support, working in quality control and scientific outreach. And now he is the director of product management, of the product management team. And like he says also, he's interested in scientific communication. I saw that he writes blogs pieces also. And I was quite sparked by the last pieces that he wrote about the state of science from Mark Timmer. And I guess I should get the book and I should read it also. So we are really excited to have you here, Dr. Perkins, and we're all here. I want to remind the audience that the questions can be posted in the chat session and at any time. And after the talk, Dr. Eric Perkins will actually take some time to answer those questions. So we turn our attention to you. We're happy to have you here. Welcome, and we're all here.
Thank you very much, and thank you for letting me talk about something that's very important to me, which is Addgene. So you already gave me a nice introduction here, but this is what I call my career map based on a plasmid map, and it has been an interesting journey for me going from my undergraduate career to Addgene, and part of the reason why Joanne thought, Dr. Kamens thought it would be a good idea for me to give this talk is because I have been at Addgene for quite some time. So I've seen it really grow as a repository. So really what I'm going to be talking to, so the first part of this presentation is going to be, well, first I'll give you a quick look at what our mission is, which is to accelerate research and discovery by improving access to useful research materials and information. And that's a very broad mission, but it's really what guides everything that we do at the repository. And I should say, as was mentioned, our executive director did give a talk earlier this year. You might see a couple slides that she shared, but I'm going to be going into a little bit of the history of Addgene, as well as what we can offer neuroscientists specifically. So Addgene was founded in 2004 by Melina Fan, Ken Fan, and Benji Chen. And our first plasmid went online, our first plasmid was requested later in that year in 2004. And so as I go through this timeline, I'm going to highlight some different parts of this. So just to show you, for those of you who are unfamiliar with Addgene, this is an example of an Addgene plasmid page. We have been compared to the Amazon for plasmids, and as in it's a way for people, you can literally just put a plasmid into your shopping cart and request it. We have a lot of information for each plasmid from the depositors, as well as information that we've that we've created ourselves in the form of next-gen sequencing of the plasmids. So for most of our plasmids, you'll see that we have a detailed map, and I'll just blow that up here. So you can see all the different components of that. And all of these components, for the most part, certainly for newer plasmids that have come into the repository, have been verified by next-generation sequencing. So that's part of that service that we provide, and the quality control that we provide, so that when we get plasmids from labs all over the world, we're verifying that they're what the depositor believes them to be. Besides plasmids, we're also able to offer, besides individual plasmids, we can also offer kits of plasmids. And for this case, I'm highlighting one that may be most relevant for this particular audience, which is our optogenetic vector collection from the Genovac lab. So all of our kit pages have descriptions of what's in the kits. Most of this comes from the depositor themselves. There's frequently protocols that come with each kit to help people understand how to use them, basically. And then, of course, there's a map of the of what the different plasmids are within the kit. So you would receive something like this in a 96 well plate format, and then you can click on either the plate or on the different links in this table to go to the individual plasmids, so you can see all that information that I just showed you on the individual plasmid page. You'd also be able to access from the kit page. And this is, we offer the things like this mostly because there are groups of plasmids like this one that you want to use in context with each other. You wouldn't necessarily be able to use them individually, so we want to offer them with all the other plasmids in context. And that's a way for us to provide large numbers of plasmids like this for a fairly low cost for the scientists who are requesting them. So after about 6 years, Addgene reached its 10,000 plasmid milestone. So that's pertinent because you'll see some other numbers coming up here. And I just added to this timeline that I started working at Addgene in about 2008, or in April of 2008. So I'm considered one of the veterans at the organization, since I've now been here for almost 12.5 years. So I want to talk a little bit about depositing scientists. So in December of 2011, we reached 1,000 depositing scientists. And that number has continued to increase. This graph goes up to 2018 as we close in on almost 40. We're almost at 5,000 depositing scientists now. So this includes very small labs, which have deposited 1 or 2 plasmids, and then it includes Nobel Prize winning labs, including 15 Nobel Prize winners. And actually, this number is probably more now because Jennifer Doudna, who's just won the Nobel Prize for CRISPR, is also one of our depositors. So I believe it's up to 16 at this point. Just briefly to talk a little bit about our deposit process, we try to make it very easy for labs. We have people, experienced PhDs here at Addgene, who can guide depositing labs through the process. It's completely free to deposit. We try to make it as easy as possible. And what I really want to emphasize is that it's a great way. There's a lot of benefits for depositors. So there's backups. You get a backup for the plasmids that are in your freezers. It's a way to share things internationally because we have a very experienced shipping team that can share things that you wouldn't necessarily be able to share as easily if you don't know how to ship plasmids to Japan or Mexico or Brazil. We have an experienced shipping team that can help with all of that. It's a way like protect losses. And for those of you who are in labs that have, when a graduate student or a postdoc leaves, which happens in every lab, you want to make sure you know where their plasmids are located. You can send them to Addgene, and then you'll always know where they are, and you don't have to dig through a freezer. A freezer like this, if you can see the one up on the right here. It's also a way to get records of all your requests for your plasmids. And finally, and this is sort of, I think, one of the biggest selling points, it's a way to get more exposure for your science. We do have data that shows that people who deposit their plasmids with Addgene, the publications that are associated with those plasmids frequently do get cited more in other papers. So it's a way to just get your work out there. And again, it's a very easy process. You enter data for your plasmids through our online website, or there's a spreadsheet you can also fill out that will help with that. And then we send you a kit, and you return those samples to us. And pretty much at that point, it's out of your hands as a depositor, other than we'll go through the quality control process and let you know if there's any issues there. So we try to make it very streamlined for depositing labs, and we are always looking for new depositors from all over the world. So in 2012, this was a big turning point for Addgene, because that's when the first CRISPR plasmids came out and the first CRISPR papers came out. And we were very fortunate to be in the right place in the right time, that we were able to start working with the labs that first started developing CRISPR tools. So within a year, we started creating our CRISPR Labs and Resources website. And I'm not going to go through all these features because that could be its own talk. But if you do go to our website and are interested in what types of CRISPR and genome engineering plasmids we offer several, an ebook, an electronic book that talks about CRISPR, and other CRISPR guides, resources, CRISPR history. A lot of people who are first getting into genome engineering, I would really encourage them to head to our website, so you can get from a very simple level all the way up to much more complex types of experiments. We have resources for all of those. This does not even cover all the different tools. These 4 things here are just the top of the page. If you were to scroll down, you would see that there are several types of CRISPR-related tools that we distribute through the repository. So this is just a couple other highlights from previous years. As was mentioned, we are a nonprofit organization, and we've been recognized within the state, within our state of Massachusetts, several times for our excellence as a nonprofit organization. And by 2014, we'd actually grown enough that we were able to open up a small branch just outside of London. So we have some scientists who work in London who travel throughout Europe to talk with scientists about depositing and also explaining what services we offer. In 2014, we had another important collection deposited at Addgene. It's our Michael Davidson fluorescent protein collection. And I know fluorescent proteins are important to neuroscientists as well, so I'll talk about this a little bit. This was a collection of over 3,000 plasmids that had been developed in a core facility at the Florida State University over many years. And it was bequeathed to Addgene and it's really one of the premier, putting us on the map as one of the premier sources of fluorescent protein related plasmids in the world. And we have a nice landing site for that, which has all the different colors that you can choose from, and you can click on any of these and go to any number of plasmids. One quick side note here, you'll see popping up around our website these blue flames. And a blue flame plasmid means that it's been requested over 100 times. So you can see that this is just the tip of the iceberg in terms of the number of plasmids that we've distributed from this collection, and it's quite popular to this day after 6 years. In 2015, we hit a big distribution milestone where we had distributed 500,000 plasmids, so 500,000 plasmids. And this has actually grown quite a bit since then. We continue to distribute very rapidly. And another milestone was in 2016, where we started reaching about 50,000 views of our blog per month. I'll give some specific examples of our blog a little bit later in this presentation, but this is a really nice resource for scientists, and it covers several different areas. I've already talked a little bit about fluorescent proteins, some very basic information about plasmids. We have a lot of CRISPR blog posts, a lot of career blog posts, and also viral vectors. And I'm going to talk a little bit more about the viral vectors later in this presentation. But this is our blog has grown considerably over the last several years, and it's been a great resource for scientists around the world. So other important milestones are our CRISPR 101 ebook, and that was one of our first big ebooks. We've since created several more, and I'll talk about our viral ebook later in our talk. And finally, in 2017, we launched our viral vector service, and I'll be talking about this as well. So that's our history up to about 2017. And these are a little bit more recent numbers up to this year. So we are up to the point where since 2011, when we were distributing about 200 plasmids a day, we're now up to around 800 a day. That took a little bit of a dip this year with COVID-19, but we're starting to get up to our distribution numbers again. We now have over 90,000 plasmids stored in the repository, almost 4,500 depositing labs, and many, many institutions that we're distributing to. We also have, besides plasmids and plasmid kits, we also distribute pooled libraries. As I mentioned, we have a couple different types of viral vectors. And as of this year, we're also distributing some of our plasmids as cloning grade DNA, which is another way which is a little bit easier for some labs. Usually we distribute our plasmids as bacterial stabs. And if you're able to order it through cloning grade DNA, this sidesteps that DNA prep process for the plasmids that we're able to offer this service for. And we always have new things in our pipeline. And so I would advise you to keep an eye on what's coming up in the next year. And as I mentioned, we are distributing to over 101 countries. And our most recent one is Uganda. And that was an exciting one, particularly for one of our senior technicians in the lab, Rachel, who's from Uganda. I know she was very excited when we were able to ship our first plasmid to that country. So we're always looking to expand to different countries to order from us. And also, as I mentioned, we're also always looking for new deposits from different countries. And that's really one of the most exciting things for me, is that we are such an international organization. And we've really touched so many different places around the world. So I'm going to pivot over to, because I am talking to Sona, I wanted to talk a bit today about the different types of neuroscience tools that we offer through Addgene. So I'll talk about some of the plasmid tools, the viral vectors, and I want to talk a lot about our educational and informational resources as well. So one of our earliest collections, and one of my favorites actually at Addgene, is the Brainbow collection. So if you can see my Zoom background right now, I have a Brainbow image as my background today. And this collection came from Joshua Sains at Harvard. And this came about when I started at Addgene, so it was about 2008. And it's still it's a great tool for visualizing neurons, and it's something that is still being ordered frequently to this day, almost 12 years later. Another collection is from Viviana Gradinaru's lab at California at Caltech. And this is where we really started looking at the AAV vectors, and when we initially got this collection, we didn't have AAV at the time, but now that that's grown, it's another great way to visualize neurons. And I'm not going to go into details of of all of these things, mostly because we have a lot of other things I want to show you, but these are just examples of some of the people who have chosen to share their plasmid tools through Addgene. Another important type of tool are biosensors. So Lauren Luger at Genelia Farms with the Howard Hughes Medical Institute has been sharing his plasmids through Addgene for several years. He initially developed his glutamate sensor almost 7 years ago, and he's actually updated it since then with other types of sensors, including this, what he calls the spaghetti monster neural labeling tool. And this is another tool. This is another way to visualize, again, neurons within brains and neuron tissues. I wanted to now go over to two of the biggest types of tools that we have are optogenetics. And I'm not going to show this entire video, but I am going to play a couple of minutes of it. And I believe I set this up so you'll be able to hear it.
Optogenetics has changed how neuroscientists study the brain. It provides a fast and precise method for controlling neurons using light. But how can light be used to control a neuron inside a functioning brain of a living organism? The key lies in a group of membrane proteins called opsins. Opsins are light-sensitive receptors that convert light into electrical signals by letting charged particles, called ions, cross the membrane as either channels or pumps. Opsins are able to respond to light thanks to a light-sensing chromophore, like retinol.
And I'm gonna stop there. But basically I wanted to just start showing that video. This is the type of video that you might want to show if you're in a lab or if you're teaching a course about optogenetics, and you just want, or you have a student that's come into your lab and you want to give a very simple introduction of optogenetics. I mean, it's kind of a playful way to show how optogenetics works and what its basis is. And I learned more about optogenetics from watching this video, and it's only 4 minutes long, but these are the types of educational tools that we're trying to offer about this particular area. Besides the fun videos, we do have some more serious things on our website, including we have a whole optogenetics section of our website, which gives links to our optogenetics guide, several different blog posts, and then you can also search for various optogenetic tools with all these different filters here, and it will give you a list of all the different types of plasmids that are available to you within our collection. And the optogenetics guide is kind of like that video that I showed is, again, 4 minutes of just a very cursory overview. But if you look at our guide, that gets into more details about how does optogenetics work, what are the applications, and just a lot more technical detail for people who are looking for something a little more advanced in terms of the information. As I mentioned, we also have several different blog posts. So we have the introduction and what we call our primer on optogenetics, and introduction on opsin delivery. And this is just to be very clear, though we do have blog posts that are written by some of our Addgene staff. Some of them, such as this one, are written by experts in the field. In this case, it was Derek Simons who talked about, he got into the details of stereotaxic surgery within this blog post. So there's a lot of really first-person writing from the people who do this research up front primarily. So it's not just Addgene bloggers regurgitating things that they see in papers. This is just a quick overview. Some of you may be familiar with some of these names, but I'm just quickly going to go through some of the different you can see some of the names and faces that you may be familiar with in the world of optogenetics. So I've already mentioned Dr. Gradnaru. Carl Dijsselroth is certainly a very important name in this field. We have a few more people here, and as I said, we have people from all over the world. And then just to give you an idea of what, if you go to any of our, what we call our PI pages or our depositor pages, so Ed Boyden is certainly a very prominent name in the world of optogenetics. And if you went to our website, to his PI page, you would see that he's deposited nearly 150 plasmids now in several different fields, but optogenetics is certainly the big one. So you have the resources at your fingertips here with the plasmids from his lab, and then you can also, if you want to take a look at his direct lab, there's always a link to the PI pages, to their own personal pages from our website, so you can quickly get there and scan through some of his own information that he puts out there for himself. So this is just a way, it's a sort of a one-stop shopping for optogenetics if you want to go through either the depositor pages or through the other resources that we have on our website. So besides optogenetics, we also have a lot of resources for chemogenetics. So like optogenetics, we have our own landing page for chemogenetics, and we have the different filters there for whatever you're looking for to help you search for the tools that you need within optogenetics. And again, we have a chemogenetics guide where you can kind of do that deep dive into some of some top level information about optogenetics, and then all these pages also have many different references to go to as a jumping off point if this is a field that you're just thinking about going into. And then if you're not sure which tool is right for you, we also have a blog post, for example, of chemogenetics versus optogenetics. Which one should you choose? Because you're going to have different, different applications. And as Maya's blog post here, there's a lot of things to think about, whether when you're figuring out which tool to choose. And so this might be a blog post for you to take a look at if you're interested in comparing the two different tools to each other as you think about your applications. One of the big types of tools that we have in chemogenetics are DREADDs, are designer receptor exclusively activated by designer drugs. So we have multiple depositors who have been involved in depositing these types of plasmids. I'm going to highlight Brian Roth, who happens to be from my alma mater. So he's deposited many of these, hundreds of these plasmids at Addgene over the last few years. And we have distributed thousands and thousands of these plasmids on his behalf. So again, this is just an example of his PI page. You can see that we have almost 400 plasmids from Dr. Roth, many of them very popular. And you can click on his lab, and he has even more resources from his lab page if you're interested in learning more about these tools. The other important thing, one of the reasons why I use Brian Roth as a way to segue into AAV is because a lot of our AAV virus, which is again a relatively new service, he was really one of the people who helped us kickstart that service to the world. So there are several types of AAV vectors that we produce at Addgene, and these include the biosensors, which I talked about, also recombinases, but more specifically for this audience, the chemogenetics and the optogenetics tools. So we have an extensive AAV page, as you can see here. And again, I'm not going to click on all of these things because I could probably give a whole talk just on AAV and these viral vectors, but I will talk a little bit about it. So if you were to go to a plasmid page that has the viral vector service associated with it. In this case, this is one of Brian Roth's DREADD plasmids. You would see that you could order the plasmid You can also order the viral vector from us with several different serotypes. And this isn't even all the serotypes that we offer for this particular plasmid, but there's several ways that you can order virus. And particularly for labs that want to try some of these tools in a variety of different types of cells, having these serotypes there is really helpful, so you can figure out what works best for whatever types of cells you're interested in studying. We also have, like the CRISPR ebook, we also have a Viral Vector 101 ebook. So this is something that I would, if you're interested in getting into viral studies, I would really encourage you to download this. Again, the articles in these ebooks, many of which come from our blog, range from very beginner, like I don't know anything about this, so what do I need to learn first? And then all the way up to much more technical tools after you've mastered sort of like, after you have a good understanding of what AAV is. We have some very specific tools and some articles about those and how you might go about using them. And this is just another example from our website of our AAV guide. And I'll talk a little bit about so I mentioned our blog. So this is from our blogger Beth Kenkel, who when she first started using AAV herself, she wrote this blog post as sort of what she had to learn for the as a first-time AAV user, which I think is really helpful, particularly for people who are completely unfamiliar with using these viral tools. Beth walks you through here. You can see the production, purification, titration. So some of those very basic things that, before you become an expert on the application, you want to learn more about just how to make these to begin with and what some of the principles are behind it. So that's a nice walkthrough there. If you're the type of person who really wants to know how your tools work, we also have some resources on that. And this was another nice post from Beth. And then we get more into some of the more technical aspects of it. So in this case, this is from one of our scientists, our UK-based scientist, Klaus, and he wrote this article a few years ago about using AAV for neuronal tracing. And then finally, we have one of our unique resources that we offer for AAV is our AAV Data Hub. So this is not something that we're able to offer, that we are able to offer yet for our plasmids or our kits. This is something that was developed over the last few years specifically for AAV and for the AAV scientists who use Addgene. So you'll see up here that this is considered beta right now. That just means that we're still sort of figuring out that we're learning more and more how to do this efficiently. And as it says up here, the AAV Data Hub is an open collection of user data intended to help scientists design their AAV experiments. And so one of the main reasons why we felt that this was an important tool to develop is because there are so many different AAV serotypes, and we don't always know what cell types are the best targets for these serotypes. So for the first time in Addgene's history, we really reached out to the scientists who ordered these viral vectors from us and said, hey, how did this work for you? And what worked, what didn't work? So we asked them to contribute data to our viral vector team and to our marketing team so that we could start compiling things to make what I feel is a really useful tool for the scientists who are ordering from us. So if you go to our AAV Data Hub, you'll see actual data from scientists, and this is frequently not necessarily published data, where they've chosen to share that with us, and you learn about what the injection site is, what the expression site is. Most of these are in mouse, but we have that there. They'll mention what serotype they use, the promoter, the tool type, and finally there's a link to the Addgene catalog item there. And then if you clicked on view details for one of these things, then you get even more information. So they have all this information. So if you about what their actual experiment was, you'll learn a little bit more about the scientist that submitted it if you ever wanted to get in touch with them to find out more information about it. And this is a service that we're really excited about growing. So for those of you who have used antibodies, you'll know that if you go to an antibody website or to a company that sells antibodies, frequently you'll see some feedback from users about what are the best applications for this antibody. You know, do you use it in a Western or what types of experiments can you use? And we're really trying to expand that concept to our data hub here. So this is something that we're actively working on growing. And I would certainly encourage anybody. We usually offer some type of reward if you do go through the effort of submitting data to us. So I encourage you to take a look at that data hub and either use it as a tool for your own experiments, or if you have data that you would like to share through the data hub yourself. Besides just being a nice way to contribute back to your community of neuroscientists, it's a way to sort of give yourself a little bit more exposure out there, so people can see your name and the fact that you are committed to sharing some of your science through this open access medium. So finally, I want to spend a few minutes just talking about, I'm going to pivot away from the neuroscience and talk about something that's also very important in the world right now. And that's just that's Addgene's commitment to COVID-19 as a resource. So we do have over 2,000 new plasmids for COVID-19 in our collection, many of which we've just gotten since March. Most of these plasmids are available both to academia and to industry. And we've had over 8,000 orders for these COVID-19 related materials in the last several months. This collection includes protocols, journal articles, and more. So we actually do have a COVID-19 plasmid and informational resource page on our website. So if your lab, if this is something that your lab is interested in doing, since it is a very timely area of research, and we recognize that some labs have pivoted over to this type of research. This again is a nice jumping off point for learning what types of plasmids might be helpful for that, some general research tools, lots of links to different articles. And as many of you know, there have been many articles related to COVID-19 that have come out just in the last half year, and also many different protocols. So that wraps up the main gist of my talk there. Addgene, I just to kind of wrap it up. I missed this picture. This picture was taken, I think, late last year. So Addgene is actually a little bit bigger than it was even taken a couple of years ago. But this is a pretty amazing group of people to work with. And not only do we serve scientists all over the world, send them plasmids and ask them to deposit back with us, regardless of where they are, but our organization itself, I don't actually know, I should have gotten this number before I gave this talk, how many different countries our employees come from. But just our employees themselves come from at least 15 different countries. And so we're very global, considering we're a relatively small nonprofit. We're a very global company, and it's been really exciting for me to work there over the last 12 years and really share not just scientific reagents with people, but also just really helping to develop this culture at our organization. So that's been really exciting. So that was a lot of information, but I would, if you have any questions about our resources or anything like that, I'd like to open up the floor to that if anybody has some questions.
Yeah, thank you so much, Eric. I'm trying to open my video here. I just don't know why the. Wait. Okay. So that was really a lot of information that you shared with us. Definitely, on different aspects also. And I guess I would like to go back to some of those. And my first question will be regarding how the scientists outside could wait. How does okay? How the scientists in Africa, because you mentioned that you were able to share the plasmid until now only to 2 countries, Tunisia and Uganda.
Oh no, sorry.
Right? Or did I
Those
got it
just happen to
wrong?
be, yep, no, those
Okay.
just happen, those happen to be the 2 newest countries that we had
Oh,
shared
great.
with.
Okay.
So we keep track of, and you can actually see it if you go to our homepage. We have a stats that are updated regularly in terms of how many plasmids that we've sent out and how many countries ordered from us. And it's just, it happens to be that the 100th the country to order from us and the 101st countries that have ordered from us were Tunisia and Uganda. But we have had other countries from Africa that have ordered from us before. And one thing that I didn't mention that I should have emphasized is, you know, we do charge a small fee to order plasmids from Addgene, and that's to help us cover our costs. You know, we have to pay all these nice people that work for us, and you know, there are expenses to the lab work and the freezers and everything. So that is how we earn revenue for the organization. But we are a nonprofit organization, and our mission is to really get these plasmids to anybody who needs them. And so what that means is if you're a lab, if you're a brand new lab or a lab in a developing country that does not necessarily have the funds, to say if you wanted to order, you know, 5 or 6 plasmids from us, but you know, that's a big expense for your lab, then then I would ask, you know, somebody who has some of those financial challenges you can always email help@addgene.org and explain what the situation is. Yep, and you can you can always email help and say, you know, this is my financial situation. I want to place this order, and our scientific support team will help you. We should be able to help you financially as far as bringing that price down, depending on what the situation is. So as far as I know, in the 12 years that I've been at Addgene, we've never not sent something to somebody because they couldn't afford it. I mean, we do ask labs that can't afford it to pay, because that's, again, how we cover our expenses. But as part of our mission, we really want to get these reagents out to anybody in the world. And that's also part of a commitment to the people who deposit with us, is they want to make sure that when they entrust, you know, giving their plasmids to Addgene, that we're going to fulfill whatever request for them, no matter what it takes, basically. So if there is a financial, I would, you know, I wouldn't want anybody to not order from Addgene because of there's a financial aspect of it. So I would just encourage people to contact us, and we'll help you out whatever way we can. One way or another, we will send those plasmids to you. You know, there are some other, particularly for African countries, there are some other challenges just for physically shipping plasmids to that part of the world.
That was my next question. How challenging it is. Like, I'm a researcher, I'm working somewhere in West Africa, and I'm interested in ordering a plasmid. from Addgene, because
Yep.
I realized you have the plasmid in your library. What is the process? Would you go in detail in what I have to do? Should I just email and say, hey, I would like to get this plasmid, and things start from there? And what are the challenges also?
Sure. I mean, theoretically, you can just place you can go to our website like anybody. I mean, you have to create, so just to kind of walk you through that process of what it's like to order something from Addgene. And I apologize that I didn't include this in a slide.
That's okay.
But it's but basically, you go to our website, you create an account. It's free to create an account, but we need to have some information about you and where you are. And you do need to, for the most part, you should be affiliated with a university to place an order with us, or some other nonprofit organization. We do have some plasmids that are available to industry scientists. So if there's any industry scientists who are watching this, you shouldn't feel excluded necessarily. Not everything is available to industry, but some things are, and more things are. But you create an account, and at that point, you can really just go to the plasmid that you want and add it to your cart, and then there's a checkout process. Basically, it's kind of like it's ordering anything online. We need a little bit of information from you. But and then payment information. And in terms of payment, there's a couple different ways to do that. We can take credit cards, of course, and then there's also purchase orders, which usually purchase orders go through whatever your
the institution, basically.
Exactly, it goes through the institution. So we will follow up with the institution on that, and then, as part of that checkout process, if you did contact our help center to ask for a discount in advance because you couldn't afford it for some reason, what they'll do is they'll create a discount code, and you can enter that discount code into the order at that point, basically, to reduce the price. So at that point, you can place your order. Now, so for the most part, when somebody places an order at Addgene, so one other thing that I did not get into details here, because it gets a little technical from a legal point of view, but one thing that Addgene does need to do for most of the vast majority of plasmids in our collection is we have a material transfer agreement. So with a material transfer agreement is basically it's a legal agreement between the requesting institution and the institution that deposited the plasmid at Addgene, which basically gives them permission to use this plasmid tool. And this is something that we've been using since Addgene's inception. It's sort of part of the agreement with the depositing institution. Like, if you're going to distribute this for us, we just want to know who's requesting this. So usually within a given institution, it's clear who should be signing off on that material transfer agreement. Occasionally, it can vary from institution to institution, depending on where you are in the world, or even within a given country, it can be a little bit different. If you're not sure who the person within your institution is who should sign off on that, then again, you can email help@Addgene, and we have a whole technology transfer team who can talk to you and help you track down who that is for your institution. So at any point in the ordering process, we have different teams of people who specialize in those different areas. So we have scientists who can help you. We have our technical support scientists who can help you answer biology questions. If you have an experiment and you're not sure whether
you should actually use this plasmid or not,
exactly,
in the first place.
Exactly. So we have a whole team of PhD scientists who can help answer science questions for you, and just figure out what order to begin with. And then we have another customer support team that can walk you through the ordering process if you need some help with that. We have our tech transfer team, which can help you with those material transfer agreements if you're having some trouble filling those out. Once that material transfer agreement is signed, basically that makes that order active and sends it to our lab, and they'll pick it for you. And that's where basically the fourth team in this process, which is we have a shipping team, comes into play. And our shipping team they're pretty amazing, actually. I have to say,
this is great. Kudos.
they have figured. I mean, our shipping manager Donnie, he's so excited when he gets an order from a country that we've never shipped for before, because every country is a little bit different. And has different challenges. I mean, some countries, we do ship most of our plasmids as bacterial stabs. So they're E. coli bacteria that have transformed with the plasmid that you ordered.
Okay, so in plates already, basically.
Right, right. So we do send most of our plasmids out as bacteria, but some countries in the world think of those bacteria as genetically modified organisms.
Yeah.
So there's a whole nother
customs
level.
Problems
Exactly
coming on. Yeah,
exactly. So we have a very long list of all the different countries and all the different customs challenges that we might have with that country. And at this point, we've seen so many things that our shipping team might get upset with me for saying this, but I don't think they're surprised all that frequently anymore, because they've seen it all. When we get that 102nd country to order from us, maybe they'll see something that they haven't seen before. But even if they did see something that they hadn't seen before, they have the tools to figure it out. And sometimes it might be a matter of, I mean, we have several different distributors that we use for shipping. So we have UPS, we have FedEx, we have WorldShip. So if you know that, in your country that, I've never seen a UPS truck in my entire life, then we're certainly not going to try to ship something to you via UPS. So we'll, if you have any concerns about how something is physically going to get delivered to your lab, then you can work with our shipping team and they'll figure out what the best distributor is to get you there. I can tell you from personal experience, because I answered scientific support questions for Addgene for many, many years before I moved into this position. Sometimes we'll get something as simple, I mean, it's not simple. We'll have somebody order something from Africa and they'll say, your package arrived and sat on a loading dock and it was, 41 degrees Celsius outside, basically. And so those bacteria just die.
It'll die.
And I can't use those samples anymore. So that does happen. And when that happens, I mean, we send bacterial stabs because for us on our end, that's the least expensive way to do it. But we do have a series of countries around the world that we know usually for customs reasons that we can't send them bacteria. So we'll send them DNA.
This is more sensitive, right? If you're sending pure DNA, then it gets more sensitive.
Well, it is, but it is more stable than bacteria.
That's true.
Yeah. So what might happen? So if somebody in Uganda, for example, were to order plasmids from us and we sent them bacterial stabs, and they opened up their stabs and the bacteria were dead, they weren't able to propagate them and purify the DNA, then they can write to us. And we would basically just resend their order for free as DNA, so that, in all my years, I did customer support for Addgene for almost 8 years. And occasionally it would take a second shipment to get something that the receiving lab could use. But I don't think we very rarely had to send anything a third time. So we've had enough experience shipping internationally now that we can usually, we'll figure out what the failure point was in the delivery process and then troubleshoot that and then figure out the next best thing to try to get your samples to you. So that's a really good question.
Well, I think it's a very interesting question for the audience, because people watching get really excited. Oh, I can have this and that, but how can I have all those things? This is always the issue. And I guess the other way around is, which usually also happened with younger fellows, they meet somebody and maybe another PI abroad, in Europe, in the USA, discuss and figure out, okay, they could use the plasmid from this PI, but this PI happens not to be one of your depositor. There are ways, well, I guess, to get the PI first to be a depositor at Addgene, because I guess the process to get the plasmid or the material from Addgene is safer and controlled than just having the plasmid in my suitcase, traveling from Boston to Kinshasa, not being sure whether I would actually be able to use it or not afterwards.
Sure. No, it's a really good point. And yeah, I mean, if that does come up, I mean, I would encourage people to, if there's a plasmid that you want, whether, it could be somebody that you met at a conference or something like that, or just from a paper. We have a lot of plasmids, but we certainly don't have all of the plasmids. But we do have a very active outreach team, and also our Sci support team does this too. So if you send us an email, and there's places on our website where you can request that we try to get something deposited, or you could write to deposit@addgene.org if you have questions about depositing. Then we're happy to reach out to those scientists and say, hey, this person would like this plasmid. You haven't deposited with us before. Would you consider it? And we'll walk them through the
process
steps and make it as easy as possible for them and explain the benefits of it. And I certainly can't claim that we can convince everybody to deposit, but usually, particularly if somebody else has requested that they deposit with us, that's frequently a very convincing factor.
It's a good
to
incentive.
Get people to deposit. Yeah, I mean,
good incentive.
I should say that the deposit process does take some time, because we do next generation sequencing. That's a 3 to 4 week process, and once we actually get the plasmids on site, it can take 3 to 4 weeks to get them through our quality control process and get them online. So it's not like you can just get this thing next week. But once it is online and once it is in our catalog, usually once you place that request, unless again we're sending it someplace that we've never ever sent to before, we can process that within 2 to 3 business days, and usually you'll have your plasmid within a week. The depositing process does take a little while, so that would be a little bit of a delay. But yeah, that's something we're getting new deposits. I think I just saw the numbers on these from our quality control lab the other day, and I think we're getting anywhere from 30 to 50 new deposits, not individual plasmids, but deposits with multiple plasmids in them, into the repository every month. So that's why we can't ever print like a physical copy of our catalog.
That's for sure.
Because we are literally adding new plasmids online on a daily basis every single day. And it changes every day. We get new things every day. And we have a whole team of people whose job it is to solicit more deposits and get new plasmids into the repository. So we're happy to help with that. And we've certainly grown in reputation enough over the last 16 years that if you do molecular biology, if you haven't heard of us, then you probably have somebody in the lab next door who has heard of us and is familiar with us.
That's for sure.
Yeah.
I guess we have somebody from Uganda asking, that's Mujib, and he's asking, who do we contact in Uganda for possibly assessing the plasmid that you sent there? Basically, which lab was it, and whether this is difficult. It's always.
You mean in terms of learning about what it was like for them to order from us?
Well, I guess he's asking which lab or where did you guys send it, because he's in Kampala, Uganda, and he wanted to get in contact with that person who received the plasmid.
So unfortunately, I don't think I can share that information because there is some privacy issues there. I could possibly, I can't look it up right now while I'm giving a presentation, but if he wants to write to help@addgene.org and say, I heard that that you shipped something to Uganda and I'd like to learn more about that process, they might be able to either point to somebody at Addgene, like on our shipping team, who knows what that entailed in case there is any customs information that they need to know about. I don't think we can divulge the exact person just because there's like privacy issues there or security issues. But we could if this person has some specific questions about what it entails to get something shipped to Uganda, we certainly have a number of people on our staff that could help answer that question.
Yeah, well, it's all about sharing and open science, I guess. And I always get very excited about this. And I guess a question to you is, what should I tell somebody to convince him that this is an interesting process to get into, to become a depositor? Because the question will come like, what do I have to win with this? What are my benefits? And so on. You touched a little bit on that, but I guess maybe you can give us 2 points that are convincing. Why should people actually go to Addgene and deposit their material?
Well, yeah, it's hard to even narrow it down to 2 points for me. But I mean, I guess one thing is just, one of the concepts that we really embrace at Addgene is, just open access science. I mean, I think we all, I tacked on to the end of the talk there, those slides about the COVID-19 plasmids. And the fact that we sent out thousands and thousands of these plasmids to labs all over the world who are trying to study this disease that is having a profound effect across the globe. And the fact that people were wanted to share those so quickly and so openly with people is, I mean, as a scientist, you should want to share your discoveries and give people the tools to use them and to grow that. So people can grow on your research. That's how science works. So from a very philosophical point of view, I think that embracing that open access is an important part of science.
Definitely.
But from a more practical point of view,
that's where we get, right?
Right. There's some monetary. One thing that I didn't mention is if you deposit with Addgene, we have a system that we call our rewards points, where if people request your plasmids, you can get points that will help you request free plasmids from us, so you don't actually have to pay
for
anything
them
necessarily. But I think the big one, and I think I emboldened this on my slide, is just getting your research out there. So when somebody searches for a particular thing, whether it's through, for a plasmid or a gene on Google, and frequently what happens is they'll be taken, if you search for a certain gene that you're studying and Addgene frequently comes up at the top of that search. And if your plasmid is one of the things that comes up within that search because you've deposited to Addgene, then even if somebody doesn't necessarily order that plasmid from Addgene, it's on their radar and they can see that paper and like, oh, I should probably take a look at that paper, and that might lead to another citation. So it's sort of another way to just, share your science at a global level and get it on people's radar and bring it up in those Google searches, basically, or whatever search engine that you use. And really, I should have shared some of the statistics from the papers that have come out, that it really does can boost the citations for your papers when you deposit with Addgene. And I've talked to people anecdotally about that as well. Some depositors who said, I published this paper in a pretty little known journal, which probably most people don't necessarily read a lot of. But after I deposited my plasmids at Addgene, my citations for that paper, tripled
just
or
explode.
quadrupled. Yeah, so that's really exciting to hear, that people are, it's just another way to share your science. And another way that that people could contribute, besides the physical plasmids, which we talked about, we do have, we publish, I don't know, 2 or 3 blog posts every week. So if you have something that you're interested in talking about, and it doesn't necessarily have to be a technical science thing, but if you want to talk about, if somebody would want to write a blog post about, this is how science works in this country, and these are our challenges, and this is how we're trying to overcome those challenges, for example. I mean, those are the types of stories that we really enjoy sharing. So if we can give people a venue for that through our blog to sort of share their story and share some experience that they've had, that's another way that they could contribute to the community through Addgene.
Wow, that's great. Really, we're over our time, but like I said, I always get very excited about open science. Maybe you can point us again to those educational tools that you guys developed, so we can find them on the Addgene website directly, just by searching the website. Those little videos and those really little technical, clear and concise content.
Exactly. So if you can still see my presentation here.
I can still see the presentation share.
So we have, you can go to blog@addgene.org. We have a lot of stuff on LinkedIn. I would actually really encourage, if you're on social media like Twitter, we have our marketing team puts things on Twitter a lot. So whenever we have a new blog post or exciting new collection of plasmids or some other educational resource, we always post those things on social media. I frequently amplify things on social media myself from Addgene, because I just think it's so exciting that we have these resources. So we do try to do that. We do have a YouTube channel, so if you just go to YouTube and search Addgene, you can take a look at that. And so that video that I shared today is
about optogenetics, for example.
Exactly. So we have that, and then we also, within our YouTube videos, we also have a lot of very simple procedures that, so lab protocols that we walk people through. Even really simple things like how to pour a plate or something like that.
It sounds simple, but it can be very complicated. I mean,
exactly. So we have a lot of those. A lot of times they're just, they're 3 or 4 minutes long. But if you just want to be able to see, some things like that are so simple that it doesn't occur to people that you want to make a video about it. But we got feedback that was like, I just want to see somebody do this so I can see it myself, basically, before I try it in the lab. So I would really encourage people to take a look at some of our YouTube videos. Our team has worked really hard on those, and they range from pretty simple to some things that are a little bit more complicated. But it's really good, at both at the undergraduate level or even like new graduate students who are new in a lab and trying some new things. There's some really interesting videos there that I think would benefit people from many different levels of science.
Wow, that's great. With those words, I really want to thank you for being here today and all the information that we received. And I am sure that a lot of people in the community will make a good use. I will myself make a good use of it. Yeah, it was really nice having you here. Thank you so much.
Well, thank you so much for having me. I had a lot of fun. And if you have any questions, always email help@Addgene, and we'll be happy to help out with any questions you have.
Sure. Thank you. Bye-bye.
Okay. All right. Bye.